Hi, I'm Vinita.
Hello, I'm Shuman.
And I'm Emily.
So today we have Dr. Emily Baer with us. Emily is currently a postdoc fellow in Dr. Alexander's lab in Biozentrum. She has completed her PhD from Department of Biological Sciences in Columbia University, where she studied genetic mechanism of neural development. Emily has volunteered in several teaching assistantships. She has received several awards for her outstanding contribution that includes Kavli Award for Distinguished Research in Neurosciences from Columbia University, International Birnstiel Award for Doctoral Research in Molecular Life Sciences. She has participated in several talks and have presented in several conferences. We welcome you to the Kitchen Lab interview today.
Thank you.
So we welcome you to present your work.
So in graduate school, I worked in C. elegans, which if you're unfamiliar with it, it's this nematode. Of course, it looks quite large in this photo, but in reality, this animal is about 1 millimeter long. And these have been really popular for research in genetics and neurobiology for decades now. Because the nervous system is quite small and it's the only animal for which we have a complete connectome of the nervous system. So that means we know every neuron in this animal and it's completely invariant, the development. So every neuron is always there. They all have names. We know all of their connections. And this is quite unusual in neurobiology. This is also a self-fertilizing hermaphrodite, which is really great for doing genetics because you don't need to be always crossing animals and keeping track of genotypes to keep kind of stable genetic lines. So those are kind of the highlights of why we love it for neurobiology and genetics. And in my graduate work, I got interested in the question of not just the hermaphrodite animal that you see here, which has been most popular in C. elegans in terms of research. But C. elegans has two sexes, like many species. And so C. elegans comes as a self-fertilizing hermaphrodite, but there are also males in the population that are generated and can mate with the hermaphrodites. And sexually dimorphic development is quite common across the animal kingdom. We mean this both in the sense of morphology. So for instance, there are many kind of spectacular birds or insects where you can instantly recognize the male from the female based on how they look because the two sexes look differently. But this is not only in terms of the way the animals look, kind of outward appearance, but this is also present in the neural circuits. And we know this because you can also see very sexually dimorphic behaviors arise, like these ducks mating where each of them will play a role. And this is something, that all animals do, especially in terms of reproduction. And so as neurobiologists, it's quite interesting to us to see these kinds of stereotyped behaviors because it means there's something stereotyped going on in the brain that regulates that. So as I alluded to, C. elegans comes in two sexes, and you can also, once you become familiar with looking at worms, you can quite instantly tell these two sexes apart under a microscope. And so the way that this works in C. elegans is the hermaphrodite has a gonad with ovaries that are fertilized and turn into eggs, and then she lays these. And then there's a male that can also mate with her. And these two sexes have different morphological features, most of which are involved in accomplishing that reproduction. And so it's not just true that you have this morphology that's sexually dimorphic, but the sexual dimorphism also extends to the nervous system. And so as I told you, we have this connectome, this kind of catalog of all of the neurons in the C. elegans hermaphrodite. This has been true since the 1980s. So that's when the nervous system of the C. elegans hermaphrodite was reconstructed. And much more recently, there was a complete reconstruction of the male connectome as well. So this just happened in the past couple of years. And in terms of the neurons themselves, what you find is that the vast majority of the nervous system, so 294 of these neurons, are shared between the two sexes. The hermaphrodite has just a few that are specific to that sex, and these are mostly involved in the regulation of egg laying itself. And then the male has several more, 93 neurons that are specific to the male. And these mostly control his kind of reproductive behaviors. And so, as I said, now we not only know these kind of anatomical differences between the nervous system, but we have complete connectomes. So we know every neuron and all its connections in both the hermaphrodite and the male. And I'm just going to very, very, very briefly, kind of in one or two slides, say what my graduate work was, which I think was to approach this from many different angles. So, ask how you get to this adult connectome, so the developmental question, and then also ask how plastic is it? So can it be regulated by things that are going on internally in the organism or externally in the environment? And then also, how this is being regulated genetically. So to just give kind of the highlights there, I found in my PhD that both genetic and environmental factors shape this sexual dimorphism. So for instance, we found genes such as this transcription factor called TRA1. This is called the master regulator of sex determination in C. elegans. It's a transcription factor that in the hermaphrodite is expressed in every cell type. And we found that it was important not only for kind of this morphological development of the difference between the sexes, but also for behavioral development between the sexes. By regulating other transcription factors, like one called DMD4, and kind of the whole genetic history of this is not as important, but it gave us this kind of genetic idea where we have TRA1 that's expressed everywhere in an animal. So this is in the whole nervous system and is important for the whole nervous system. And then it's controlling much more specific genes. So we found that this transcription factor was only expressed differently between the two sexes in just 2 pairs of neurons in the whole animal, which is a thing that's really great about working in the worm, is that you can really narrow down to very kind of specific biological decisions and figure out what's controlling them. And so we found that this was differentially expressed between these 2 neuron pairs in the animal. These are important not only for shared behaviors, so behaviors that both hermaphrodites and males do, but also for mating behaviors in the male. So these develop very different functions between the 2 sexes. And we found that DMD4 was required in the hermaphrodite, but not in the male, to kind of control the way that these neurons connect to their downstream inner neurons, and then control some of these behaviors that we were interested in. And then one of the earlier studies for my graduate work, and this one I think was kind of the most surprising one to me. So this kind of genetic stuff, we had an idea what we were looking for. It's not a revolutionary idea that genes are involved in development. We knew to look for them, and we wound up finding them. But another study in my PhD, I think, happened quite accidentally, which was one where, and the details aren't all important here, but we found that basically in a male animal, whether or not they are exposed to a period of starvation, so whether or not they have access to food via serotonin signaling pathway, can also impact the way these same neurons are wired to one another and thus impact behavior. And so this one, I think, was a lot of fun, because it was one of these things where you very much have to let go of the idea that you're smarter than the way that biology works and just kind of let it happen to you. Like, I wouldn't have guessed that this happened, but here we are. And then you just have to kind of do your best to follow it up and study it. And so I completed my PhD at the end of 2019. And at that point, I moved to my postdoc. I'm going to give you an extremely kind of brief and vague outline of what I'm interested in my postdoc. I'm not keeping secrets or holding anything out on you. I think at this point, there's also a vague understanding of this project, which is quite normal of a project at the beginning. We don't always have all these glamorous stories to tell yet, but hopefully they're coming. Oh, and I'll tell you one more thing about sex determination, just to try to loop this into a broader context and outside of, like, do we only care about this in nematodes? Is this only relevant in worms? And I think this is quite an interesting topic generally, because if you look at the way that life has evolved over time, is that, you have species that produce asexually, and these are located kind of here on the left of this tree in bacteria. And then about 1.2 billion years ago, sex evolves, and you have this massive expansion of species, right? So like everything to the right of this giant sex evolving event is what happens once you start reproducing sexually. And this gives you just a ton of opportunities for kind of evolutionary differentiation and stuff like this. So as an evolutionary concept, it's been extremely successful. And I think it's a really, really interesting thing to study. And of course, I studied it in nematodes, which are here on this evolutionary tree and just represent one tiny branch of this. But it's certainly been important for all of these species. So to continue this theme of branching out on this evolutionary tree, from my PhD to my postdoc, I switched from working in C. elegans to working in zebrafish. Here I'm just showing you the embryonic development of zebrafish. And I think this is another genetic model organism, if you're not familiar with, one that's been used for a bit less time than C. elegans, but I think is great because you know, one of the things that's most amenable, I would say, about zebrafish is that all of this embryonic development you're seeing occurs externally. So you're not, you know, trying to get embryos out of a uterus of some mother or something, which can be very difficult to study development. And also, much like C. elegans, embryonic and larval development of zebrafish is completely transparent, which is amazing for microscopy and also amazing for seeing things happen. So yeah, in some ways this represents a big transition for me, but in other ways, I'm just sticking to animals I can see through and hoping it works out. But one of the things that is also worth noting in kind of comparing something like C. elegans to something like zebrafish is there are people who will call kind of the nervous system of C. elegans, they have this collection of neurons in the head. Some people call this a brain. This is controversial whether or not it's a brain. And so the worm you can kind of think of as having a very organized nervous system, but it's like one unit almost. And the thing that kind of sparked my interest about zebrafish is that you have this centralized nervous system, but it's still, you know, it has to get all these signals from the body. And I think we've thought for a long time about how the brain processes sensory feedback. And I mean this in terms of what we're seeing and what we're smelling, and what we're tasting. But your body's not only getting sensory feedback from the outside world, you're also getting sensory feedback from inside of yourself. And this is really, really important for you to survive. But I think we maybe don't think about this as much. And this is just an image from a recent editorial in Science magazine that's credited here, pointing out that there's this two-way communication between the brain and the body. This is the vagus nerve, which is one of the cranial nerves, so the nerves in the head. In humans, it connects to a part of the brain called the medulla oblongata. In zebrafish, it's in the hindbrain, which, you know, is just the back of the brain. But what this does is it connects to every organ in the body, and it takes sensory information from all of them and sends it back to the brain. And I think there are a lot of interesting questions now about, you know, what are brains doing with this information? So if I kind of reduce it to the most simple approximation, you can think of a brain as something that takes a stimulus from the outside world, and then behaviors come out, right? It's like you put stimulus in and then a behavior comes out. But the brain is not only getting stimulus from the outside world, you're also getting sensations internally. And one of my interests is whether changes in internal sensations, so changing what's going on inside the body, can make the brain take the same stimulus and then output a different behavior. So that's what I'm kind of interested in doing in my postdoc. And yeah, hopefully in a couple more years, there will be a lot of stories to tell about this as well.
Thank you so much, Emily. It was an amazing presentation and it was so exciting, and also at the same time, very well summarized. So no two people have the same story of falling in love with science. And we would like to hear your story of how you fell in love with science.
Yeah, from kind of a storytelling perspective, it's quite convenient because there really is like one magical moment. So as a student, I was always kind of naturally drawn to biology and good at it in school. But I came from a kind of background where no one was an academic, no one was a scientist. I didn't know any scientist. I'd never met anyone with a PhD before I went to university. And so everyone said, yeah, well, you're good at biology, you'll become a medical doctor. And I was like, okay, that makes sense. That's what I'll do. That's fine. And so, you know, I went to university and I was majoring in biology and I was like, I'm going to become a medical doctor. That's the only thing I know that you do when you like science and you know about it. And I got to my third semester of my undergraduate, and I was taking some biology class. You know, I'm just kind of at this point where you're transitioning from the introductory coursework to kind of more specific stuff. And one of my professors said, you know, tonight there's this poster session on campus, and it's where the other undergraduates are going to present what they did, you know, like in a summer research program on campus. So all the undergraduates that did summer research are going to present their posters there. I didn't know what a poster session was, but she told me that, like, as my homework for this class, I was supposed to go to this poster session and I was supposed to write one page about a poster that I saw. And I was like, okay, I'm just doing my homework. I'll go to this poster session. And I don't, thinking back on it, it's kind of funny how naive and accepting I was. Like, I didn't understand. I didn't know what graduate school was. I didn't know what a postdoc was. I didn't know what research was. I can't even guess how I thought my professors got their jobs, because I had no understanding of anything they had done. And I was like, I don't know, like, maybe it's just a special kind of teacher. Like, I don't know, like an elementary school teacher and a high school teacher and then a college professor. They're just all kinds of teachers. It had not occurred to me that these people had done any research or gone to graduate school or anything. So I go to this poster session, and I'm talking to these students and I'm looking at all these posters, and I realized that my professors also had research labs. And I realized that this was a job, which seemed very suspicious to me. I was like, this can't be a real job. People get paid to do this all day long. And I'd never seen, you know, what a poster session is like. There's like pictures of cells expressing GFP and like development happening. And I thought I was asking for some secret information. I go up to one of these students and I'm like, how do I get this job? And they're like, oh, you just email a professor and you ask them to research. I'm like, so I just email a complete stranger and tell them I want to research. They're like, yeah, do that. So I go home and I do this. Yeah. I go on like the department website and I see all the research topics about which I know nothing, but just choose one that seems kind of fine to me. And I emailed this professor like within a day. And so, yeah, I would say like from going to this poster session and finding out that science was a job to joining a lab, less than 1 week elapsed. And I just decided, I was like, well, this is what I'm going to do. This is my job now. And it's been that way ever since. So, yeah.
That's a fantastic story.
that sounds well. Coming to our next question, the cool part about discoveries are it can happen, like it can be good and bad in both the ways, but it helps people understand who they are, like yourself, like the better you. So off-bench discoveries are often neglected but has a very huge impact in our lives. So what was the best or the worst thing you discovered about yourself in the journey of your PhD and postdoc?
I think I remained and have remained very kind of convicted of the idea that like, this job is what I'm doing, you know, this is just for me. And I've just kind of found, I think that many people have a thing that's right for them to be doing in their lives, and like you have or haven't found it yet. And I feel quite lucky to have found that thing for me at what was relatively a young age. This can both be extremely empowering and very depressing knowledge. Like on your good days in science, you're like, yeah, you know, this is the job for me and it feels great. And then you can have a really crappy day and you're like, oh, I cannot believe that this is the job that's right for me, right? But I never kind of think that it's the wrong thing to be doing. Sometimes it's just like, this is what I got, come on.
So PhD is definitely a long journey. And one of the things that happens is that we also realize a lot of things about ourselves, and we tend to incorporate a lot of new things. So how has PhD helped you develop your personality and your philosophy towards science and life in general?
I started my PhD when I was 21, which is a harrowing age for anyone, I think, just kind of developmentally. I'm very aware that for the first 4 years of my PhD, my brain was still not finished developing. And so I think that whether or not I'd been in graduate school, I would have changed a lot over that time. One of the things I alluded to in my presentation was that one part of my graduate work, which was this stuff on looking at how starvation affects the way that the sexually dimorphic circuit developed. Really, it was a discovery that I stumbled into a bit accidentally. And it was doing this study. It didn't seem like it was working out because I was the smartest or I had all the right ideas. It just seemed that this was something fundamentally true that was happening in biology. And I was just racing to keep up with it. And so I think one of the things that I take quite seriously now is that I'm probably not smarter than the science. This has been true for hundreds of millions of years, and it's just up to me to kind of tell the truth about it. And so I think and in science a lot, I think we use these kind of what's called the lone genius narrative, right? So-and-so had the best idea, and then they learned this fundamental thing, and then they got a Nobel Prize. And we'll never know if it had to be them, right? Would someone else have done it the next year? We can't kind of do that controlled experiment, but I think my graduate work kind of, disavowed me of this kind of idea.
So failures have never been easy to anyone, but with time we understand that how to deal with them, and everybody has a different coping up mechanism. So how do you deal with your failures?
There's different kind of severity of failure. And I think that this is one of the things that people just starting out in research often have to get used to is, I don't know, if I'm mentoring an undergraduate or a junior graduate student, they'll come to me very nervous because their PCR didn't work or something. And I'm extremely nonchalant about this because it doesn't work a lot of the time. And this kind of failure, I kind of thrive on, I would say. One of the things that I tell my students a lot is the difference between me at the beginning of my PhD and the end of my PhD is not that I make any fewer mistakes. It's just that I know how to fix them fast enough that no one notices. At least for me, maybe some people have developed some bulletproof methods, but I think I just figure out how to troubleshoot the mistakes faster. And so I can, I have a better instinct for what went wrong. And this I think is kind of fun to do because, my PCR doesn't work. And this is a problem, but it's a problem that I know 7 different ways to try to fix. And I might have to try all 7 of them, but eventually I'll get it. Failure in science, I kind of enjoy because there are all sorts of big problems in the world that I have no control over and I can't do anything about. And this is really hard, but a failure that happens at the bench, I can always fix. So I don't know. I kind of enjoy my scientific failures.
Nature or neuron is a dream of many young researchers. You have not only achieved that feat, but you have maintained the level of excellence. So what are the most essential elements one needs to incorporate in themselves to make their dreams of doing excellent science come true? And also, do you think that publishing in a big journal would make that science more worthy?
My Nature paper was also rejected somewhere before it was published there. So it's not like a home run that I hit out of the park instantly and this was all good times. This is the study that I was talking about that I feel very particular about because it wasn't all my own personal great ideas and kind of an accidental discovery. And I think it's really not, I have this paper and then I have papers that are published in lesser journals. And I don't feel differently about any of these studies. It's not like the Nature paper is my favorite child. So I think to me, the most important thing is, am I telling the truth about something that's going on in biology? And if only 5 or 6 people care about it, I don't think it's any less worth telling. And like, I think I owe it to them to put it somewhere that like they can find it and it will be useful to them. So I think a lot of this about, you know, what we're doing at the end of the day when we're publishing is like making our discoveries available to other people. And they're available to people when you publish them in Nature, and they're also available to people when you publish them in other journals. But that's the most important part.
A close friend of mine had once referred me to an article known as The Mundanity of Excellence. So it spoke about how talent is more about persistence and practice rather than something that you are born with. And it is through practice of certain apparently mundane habits that one achieves that excellence. So through the process of your scientific journey, for the sacrifices that you made to keep yourself in these mundane practices that helped you achieve your goals?
There are immense sacrifices that come out of this job. You know, my transition from PhD to postdoc, like no one was forcing me to move to a different continent where I knew nobody and abandon all of my friends and all of my loved ones and everyone I knew. And yeah, kind of my native country, but this was a choice I made because I was interested in like, yeah, taking this angle scientifically. On the other hand, as you say, kind of the mundane practice of things that lead to excellence. One of my hobbies, and perhaps the one I spend the most time on, is also one that people think is extremely kind of masochistic, which is endurance running. So I run marathons.
Wow.
And yeah, but running a marathon, like, is one kind of glorious or not glorious race that you do on one day. That's like 4 months worth of running 4 days a week before that. And this is something that I structure a lot of my day around. It fits very nicely into a scientific day.
Though there cannot be one single recipe to it, but we would like to know that what do you think is the most special ingredient to become a scientist? Like, is there anything specific you can think of?
I think, to me, I always boil it down to like, you just have to want to do it. And I think you really can struggle if you haven't thought through why you're doing something. And this is true of kind of science as a career. And also it's something you always have to be mindful of at the bench. If you're not sure why you're doing an experiment, it's probably not worth your time to be doing it. And in that sense, this career and like your whole life is an experiment that you have to be weighing of why you're kind of spending the time on it and why you're spending the effort on it.
So for people who are outside the scientific community, they find it very hard to conceive how research on worms can help humans in any way. However, we know that it is an excellent model for answering fundamental questions. So could you share the excitement of working in worms and the potential that it holds, especially for frugal science?
I think that there really have been great discoveries that have come from the worm, in the sense that the amenability of it to genetics, for instance, the worm was the first animal to have a complete genome sequence. And I like to use this example because I think the genomics projects, like the human genome, is that, you know, we said, oh, we're going to get the human genome and then we'll understand everything and we'll cure every disease. This was not true. And I think disappointed a lot of people, and it made them ask, like, what's the point of doing this? But no one can imagine doing science now without the genome technologies that we have. Like, we sequence genomes so casually now. And we only do that because we developed all the technology starting with bacterial and yeast genomes, and then doing the worm as the first animal, which enabled us to then move on and do like larger and larger genomes. And so I think in that way, I'm very hesitant to overpromise, but I think that kind of in the act of doing it and just trying new things. And so I think it's, kind of in these, as you said, kind of the mundane aspects of excellence. So really kind of practicing these things that don't seem like they might be the most important thing in the world when you're doing them, but the results can be really surprising.
Humans hold the power to change whatever they want to. If you were given a power to change something, what would that one thing that you would like to change in academia?
I think that one thing that's really striking now is that there's a huge generational difference in terms of people who are hoping to pursue academia as a career now versus 50 years ago or 30 years ago or even 20 years ago, which is that it's become much, much more competitive, right? There are more people trying to get an ever fewer number of jobs, kind of this extreme scarcity in terms of how people feel about their careers. I don't think that you do your best science, and I don't think that you bring your best self to your work if you're always in kind of a state of existential panic of whether you can continue in your field. The thing that I would change is that people would feel less that they're competing for very, very little resources in terms of jobs or money, not just because I think it'd be better if the jobs or research money existed, but because I think people would, kind of show up in a better way to their own work if they weren't in a state of panic.
So you have traveled a long way in academia. You were mentored by several, and you had the chance to mentor several people. How was your experience as a mentor and a mentee?
Yeah, I think this is it's very interesting. And this has also been interesting to me to make the transition, I think, from being a graduate student to being a postdoc. People kind of told me that this would happen, and I don't think I understood it until it did, is that one of the things that's particular, well, it's not particular to science, this is true in all aspects of our life, generational and the way we act in the world, is that there's kind of like a hierarchy system and like a power system that exists. And not just in the way that you treat other people, but also in the way that you're regarded. And what I mean by that is there's kind of more professional distance that exists as a given between me and a graduate student, or me and an undergraduate student now, versus when I was a PhD student. And so one of the things I think, mentoring is part of this job, and it's a skill we have to practice. And I think the hard thing of it is much in the same way where there are days that you forget the polymerase, right? And your PCR doesn't work. Like there are days I just don't show up as the best to mentoring. And I have to remember that my mentors also will have days where they do the mentoring equivalent of forgetting the polymerase. Like they're just going to fail at this part of their job some days too. And so I think the thing for me that is important is to just always kind of assume that everyone's trying their best. Like I just don't come to the work with the assumption that like, well, my mentor probably wants me to fail, or like they have secret malicious feelings for me. I'm just going to assume even if something goes wrong, it's just a day that they like forgot the polymerase in their mentoring. Yeah. So I think of mentoring as one of the many skills we need to learn, but yeah, the most dangerous one because the recipient has feelings, unlike my PCR.
So traveling away from family and friends to pursue dreams is not easy. And so how was it to move from US to Switzerland? Do you have any advice for international students?
I think advice that I would have, I think it's certainly, I don't know that even though it has been more difficult in a lot of ways that I expected, I also don't regret my decision. And I think it's good, even if I was surprised by how different the environment is and how different the academic environment is and how much that impacted kind of the way that I feel and the way that I work. It's all kind of data, and it's all good to know. And it's good to know and understand the way that things are run differently in different places. Like one of the things that is just unbelievable to me here, and I don't understand, is the institute where I work, like, does maintenance on the computer system for weekends, like once a month or once every other month. And then they turn off like all access to your data, all access to the email. And sometimes I just laugh because I imagine it happening even a single time when I was in graduate school, and how upset everyone would be to have a weekend where they didn't have access to the data or like couldn't send any emails. It would be just a catastrophe. It would be a huge deal. They would put it in the student newspaper, and here everyone just kind of accepts how it is. And of course, that directly impacts the way that I work. I also don't have access to the data or the email for a weekend. But it's just fascinating for me to see people react to that in a different way and not have it be the end of the world to them, apparently.
So now we come to the last question. What do you think about Kitchen Lab?
I think it is so cool. As we talked about earlier, I somehow managed to go through my entire kind of childhood and the beginning of my college experience, like not knowing anything about science or what it was. And I think it's amazing to me that you've managed to, just looking at it, like it's a real lab, right? That's a real molecular biology lab. And just
no, it's a behavioral lab actually.
Okay.
We don't do any molecular stuff.
But yeah, kind of have this idea that you can be doing real kind of experiments, and you can just kind of do this anywhere, which I think is an idea I don't think about enough, which is like, yeah, it's actually quite possible to make all of this really accessible. And I think it's really unique. And I think it's a thing that there needs to be more of. I know that when I was in New York, they had something called the bio bus. And so they put this lab in a bus and would just like drive it around the city, and then students would come and see.
Wow.
And so I think all the ways that we can recreate labs, and not just, we talked about science communication. And I think I was thinking of it then in terms of just talking, but talking can be very boring. And sometimes I think it's all the better to be able to really show people what it's like. And yeah, that's where all the magic stuff happens.
Thank you.
Thank you so
much, Emily. It was great talking to you.
Thank you for listening. Stay tuned for more videos.